Dr. Thomas Koch is a researcher at the Ontario Veterinary College and has been working on regenerative therapies using stem cells. Welcome, Dr. Koch.
Thank you, Jackie.
Can you briefly describe your research project?
Yeah, so the project we're talking about now is about what we call vitrifying cartilage. So it's basically a freezing method to preserve cartilage long-term in liquid nitrogen storage. But the process to freeze down this tissue is called vitrification because it's a rapid freezing process where you avoid the water within the cell and the tissues forming small crystals that would cut and disrupt the tissue in the freezing process. So it's a unique method of freezing tissues where you preserve the tissue structure. And it's well known for the freezing of embryos, for example. But it has been very hard to freeze down cartilage just because cartilage is a unique tissue that has a lot of what we call extracellular matrix, so components outside of the cells, and it's negatively charged, so that makes it technically very difficult. But my collaborator, Professor Nat Jomha in Edmonton at the University of Alberta, has developed a method for this where they have shown this to work in humans and in pigs, and they have protected this method. So we are collaborating now on how to freeze equine cartilage.
What are the potential practical applications of this research for the horse owner?
So the practical implication is that, as we all know, joint disease is one of the main causes of early retirement of horses, where they don't get to fulfill their athletic potential, and there's lots of cost associated with that, both financially, emotionally, and so on. And a lot of these joint diseases are due to a localized defect on the cartilage surface. So that is really what this project is targeting. It is localized, what we call focal cartilage defect. So it could be the size of a toonie or something, but if left untreated, it can progress and form osteo-arthritis, which is a disease of the whole joint. So finding better methods to treat these localized cartilage lesions would allow us to extend the athletic career of these horses and maybe delay the onset of osteoarthritic changes.
And of course, there'd be a practical application to being able to cryopreserve this stem cells, right?
Yeah, so cartilage transplantations in different forms have been done a lot. The problem is that if you do it from the patient itself, then you need to harvest cartilage or cartilage and the underlying bone from another area of that patient, typically within the same joint or a related joint, but you're basically going into an area where there's no injury, and you're now harvesting a piece of tissue in an area that is deemed to be less critical because maybe it's not directly weight-bearing and so on. But you still run the risk of creating injury in that healthy tissue site. The other thing is that when you transplant that cartilage, it comes from an area that has not been weight-bearing. So it's typically thinner. Biomechanically, it's not as strong as the area that you're trying to treat. So that's complicated. Another method where they take tissue from the patient itself is that you go in, you harvest cartilage pieces, you take it to a laboratory, and then you spend 2 or 3 weeks isolating the cells from the cartilage, so it's called the chondrocyte. And you can then expand that in the laboratory so that you have lots of them after 2 or 3 weeks. The problem with that is that during that growth phase in the laboratory, the cell changes, so it's no longer really a chondrocyte when you have a lot of these cells. Secondly, you have to ask the patient to come back again 2 weeks later, which is inconvenient, and then you inject these cells. However, it has proven to be efficacious in humans, but in the equine industry, it has not been used very widely because it's very time consuming, it's very expensive. So the alternative is that you try and look for a tissue that's not from the patient itself. So, and that has been done as well, both in human and in equine medicine, where you can take from a cadaver limb, a horse that has died for other reasons, but the cartilage of that joint is healthy. So you can harvest that and then transplant it. But again, logistically, it's complicated. You have the risk of disease transmission because it's between two individuals. But interestingly enough, it has shown that you can take cartilage from one individual and put it into another horse without a lot of immune rejection. So it's difficult, it's cumbersome to do it because you need donors. There's a lot of logistical processes that needs to be in place to do this. But from a biological perspective, it seems possible that you can take cartilage from one animal and use it in another without it being rejected. So we kind of thought of all of these challenges, and then we said, we know that the stem cells we have in the lab that we can freeze down, we can thaw them, and we can in the lab direct them to form cartilage tissue. So we have a source of cells in the laboratory now that we can use to build cartilage tissue. So this will not be from the patient itself, but we need a way to store it now so we can put it into patients. And that's where the work by Professor Jumha is very interesting, because that seems to be the missing piece, where if we can vitrify this cartilage now, we can then store it long term. And then when the surgeon has a horse that they would like to treat, we can thaw that cartilage tissue that has been generated from stem cells, and then the surgeon can use it right away on the day of the surgery. So we avoid the harvesting from a healthy area of a joint. We avoid, multiple visits. We have to harvest, bring the horse back, and we can control the risk of disease transmission much better. And we can also make sure that the thickness of the cartilage and everything else is more appropriate for that area that we're trying to heal.
How has this exciting injection of research funding from Ontario Equestrian helped move this project forward?
Yeah, so this money obviously from the Ontario Equestrian is critical to us, because we have been able to do some preliminary work prior to applying for the money, where we took equine cartilage from cadaver limbs and we shipped it to Edmonton, and they showed that their protocols that works for human and pigs seem to also work fairly well on equine cartilage. But this was not cartilage from stem cells. It was cartilage from horses that had been euthanized for reasons unrelated to this. But it showed us that the protocol would probably work on equine tissue as well. But as you know, working with stem cells and doing this laboratory work is expensive. So the question we now had was, would we be able to freeze down the stem cell, produce cartilage? And this is a relatively small project, but it's a very important project because it will allow us to answer this question. And that's basically the only question that we have. Are we able to vitrify cartilage from equine stem cells well? And if once we have answered that, we can then apply for funding to build on this. And the other critical part of this is that the horses and humans actually share these conditions of cartilage, and the horse is recognized as one of the best models of human cartilage disease and also repair. So we will be using the data from this project to apply for funding both human health grant using the horse as a model, as well as equine specific grants for the sake of treating horses. So our focus is on treating horses, but we will also use this project to source money from human medicine with the argument that anything we can make work in the horse is likely to also work in humans.
Certainly some exciting applications. What are the top take-home points you feel that are important to the industry for the care of people's horses?
I think number one is that there's a big group of people that are benefiting from Equine Guelph and their research, and it's not only me, but there's a big group of people here dedicated to doing research for the betterment of equine welfare. And you know, it's a community effort of everybody, us as researchers, but also people in the community supporting our research. And then the other message is that, you know, we're targeting a condition here that is very common in the equine athlete, and that is across species, it's across disciplines. Every horse has a high risk of a joint problem and a high risk of a local cartilage defect. Either it's by developmental disorder like osteochondrosis, or whether it's trauma following exercise and athletic activity. I think those are the main take-home messages.
And in closing, Dr. Koch, is there anything you'd like to emphasize, sum up, or glance into the future of regenerative therapies?
No, I think it's a very exciting area. My lab has focused on cartilage repair, but we are focusing on other areas as well. One area that we are taking an interest in is that these stem cells also seem to be able to treat bacterial infections. There's been reports in dogs where they had long-standing multi-drug resistant bacterial infections that had been treated for months and months without success. And then when they added stem cells to the treatment protocol, 5 of 7 dogs were completely cured, both on clinical signs but also on bacterial culture. So I think the area of bacterial infections are very, very interesting. Other inflammatory diseases are interesting. I know that there are trials going on in Canada and elsewhere in the world treating COVID-19 patients with similar stem cells for their anti-inflammatory properties. So I think we'll see a lot of pure cell-based therapies developing. The other side is that a lot of these cells exert their function by secreting different factors, and we can actually capture these secreted factors from the cells because they are secreted into the culture media that we grow the cells in. But there's a whole industry developing around what the cells are secreting, where you may not end up using the cells themselves, and logistically that can potentially make things easier because you don't have to freeze cells. You can have a more traditional type drug formulation.
Dr. Koch, thank you so much for being part of our interview today, and we look forward to future updates on your research.